Okay. Good morning, everybody, and welcome to the third meeting of the SEALS. It's the SEALS Cafe, which are these two boys X-ray dialogues organized by our society. And today I'm very glad to switch from last time we were talking about Alzheimer's disease, and today we have two guests. They will speak about vaccines. This is a very important field of research. And again, the two voices, this dialogue is between two people, one from the field, which is Fabrizio Chiodo from CNR, who studies vaccines also from pharmaceutical point of view, and Vincent Olieric, which come from Synchrotron fields, and which will propose some tools, important tools, combining crystallography and synchrotron radiation to study the same subject. So I immediately stop my screen, and I give the talk to Fabrizio.
Yes, let me share the screen. Okay. Okay, can you see everything?
Yeah, perfect.
Okay, perfect. So, well, hello to everyone, and thanks to let me play this cafe idea with the colleague. And first of all, I'm very sorry for two reasons. First of all, because I misunderstood the length of the talk, so I have to fix it a few hours ago. But this is not the main problem. And the other thing is that I have the feeling that I am a little bit too far from your field of research, but it's also from one point of view also very challenging and interesting for me to show what we are doing in the field of vaccinology, and today it will be mostly on SARS-CoV-2. So sorry if the language is not the appropriate one. I will try to be very basic, but I think it will be also very interesting to establish some collaboration. But the most important thing is to trigger your curiosity by talking about carbohydrate-mediated consideration in designing vaccines. So I do consider myself and most of my co-workers carbohydrate scientists. We have nothing to do with diet, or we have nothing to do with diabetes, but we like to study how host pathogen interactions are affected by carbohydrates. Because any living cells, but also viruses, are totally surrounded by a layer that we call glycocalyx, and a layer of carbohydrates. So evolutionary nature have used this code to talk to any field to any signaling system. So you see, this is an epithelial. This is just a vessel, no? To make it easy. And this is just the sugar coat that is really coating the vessels. Here on the right, you see our red blood cells, because each of our red blood group cells, A, B, O, we are talking about carbohydrates. And also here you do see two bacteria that are staining in a different way, and in both cases you can nicely appreciate this layer of carbohydrates. So you can imagine that anytime host and pathogen cells are talking to each other, but also in the tumor microenvironment, for example, from one side you are going to have these carbohydrates depicted here in this way, but on the other side you can have receptors, innate receptors, something that nature has conserved to recognize this carbohydrate, to find a signaling from these carbohydrates. And remember always, these are kind of parallel situation where simultaneously the host can have glycans and a receptor for glycans, and the pathogen can have receptor for glycans and glycans. So this is how when I teach glycobiology to students, this is always the main focus we have. So why am I discussing this type of things if I'm supposed to trigger your curiosity by talking about vaccines? So first is also to have a more complete vision that there are no scientific fields where carbohydrates do not involved. So I mentioned cancer, I mentioned infection, and I will mention immunity. But remember that all the interaction in nature, and keep in mind that already DNA is per se a glycoconjugate construct. So can you hear me?
Yeah, okay, now it's fine.
Because I had an echo from somewhere else. So carbohydrates are involved in any. Everything is blocked now. Can you hear me?
Yeah, we hear you.
Okay, everything was kind of blocked. So any interaction in science are in dealing with carbohydrates, and as I said, DNA, for example, is a pretty famous glycoconjugates. So today, just look at the right side. This is what I will try to uh, analyze with you. So on the right, you do see the very pretty famous spike protein of SARS-CoV-2. This is the way carbohydrate scientists like to think about this type of protein, because any protein are mostly covered by carbohydrates. And as I mentioned, these carbohydrates are extremely important from a structural point of view, but also they really do mediate the first preliminary interactions that our body are feeling when we have to combine host pathogen interaction. So when we have to design vaccines, on the left I had the example of Streptococcus pneumonia that I have to skip. But when we have to design vaccines, we must consider always carbohydrates in our vaccination design. So, but viruses are pretty interesting because any living cells, in most of the cases, we have a machinery of glycosylation. So our cells are able to cover themselves and their protein with a machinery, enzymes that are able to put carbohydrates on their structures. But viruses, no. Viruses are far from a living system per se. So viruses, they do use our machinery, the machinery of the host, to cover themselves with carbohydrates. And there are pretty famous viruses where glycans are pretty heavy on the surface. And I'm just going to mention HIV, GP120 glycoprotein, that is most of its molecular weight is actually carbohydrates. And this is maybe one of the key reasons why it is extremely difficult to develop vaccines against HIV, because the access to antigen are really masked by the strategy that some of the viruses have used to escape from our immune system. So luckily, beta coronaviruses like SARS, they are also glycosylated, but fortunately, well, let's say at the moment, fortunately, they are not as covered by carbohydrates like other viruses. So yes, viral proteins are glycoproteins. So yes, these glycoproteins are talking with our immune system, and if we have to design vaccines against a virus like SARS-CoV-2, we do have to consider carbohydrate-based consideration. So this is another way of depicting the famous spike from SARS-CoV-2. On the top, you do see the receptor binding domain. This is the S2 region, and as you can see, the spike protein is fully covered by carbohydrates. I'm colorblind, but I think this is kind of a purple color or whatever it is. You're gonna appreciate that this is really how the protein is covered by these carbohydrates. And I have this video here. Let's see if I can let it start. No, because here I can hide. No, I cannot let the video go. No, try again. Okay. So this is actually the way the full spike looks. This is a model that was done from the colleagues in US at the beginning of the pandemic. So you do see you have it's a trimer, and you have this 3 receptor binding domain on the top, fully covered by carbohydrates. But what is extremely interesting from our perspective is, and you're gonna see when the video will go to the end. Now you're going to see it's a very dynamic situation where the receptor binding domain is mostly going up and down here. And this is extremely important when we have to design vaccines. So the vaccines I will describe are all receptor binding domain based. They're authorized in Cuba, and not only in Cuba, but also in other countries. So they're all receptor binding domain, and why it's important to discuss about receptor binding domain based vaccines and to talk about carbohydrates, because was extremely I have troubles here again. What is extremely important to realize is that when the receptor binding domain and actually all the full spike is in its close position, part of the antigens are completely hidden from our immune system. This is actually how the vaccine from Pfizer and Moderna are working, but also Novavax. So they do present in somehow the spike protein when it's closed, and then at some point when ACE2, that is the host receptor, is approaching, the receptor binding domain, look the progression from A to E, is opening, leaving part of the antigen exposed. And there is also a very important glycan here from another receptor binding domain that is pushing this receptor binding domain up. So this structural consideration were extremely important when we did decide first to work on receptor binding domain based vaccines, because we did think that the accessibility of the antigen was more important. Second, we expressed the receptor binding domain in a mammalian system because we work on carbohydrates, and we do think that this type of keeping the original glycosylation was extremely important. Actually, now we know that most of the broadly neutralizing antibodies among the variant of interest of SARS-CoV-2 are nicely recognizing these carbohydrates in position 3, 4, 3. So we developed different receptor binding domain vaccines and expressed it in a mammalian system. There are also considerations that I don't have time to mention why we did select the receptor binding domain and not the spike, because we didn't want to interfere with other branch of our immune system. So I work since 2014 for and with the Finlay Institute in Cuba. I'm professor in Havana, also the Faculty of Chemistry. And this is an institute that has always been involved in the development of vaccines, especially for pediatric vaccines. And I will go on it. So the idea was to use technology that the lab has been used for a long time, but also to be sure about the safety of this product. So I will start with this product that actually was developed after, but is to let you understand the rationale behind our decisions. So Soberana Plus is a dimer of the receptor binding domain with a very interesting disulfide bridge here, expressed in mammalian cells. And we developed this first for the convalescent population, because the idea was, okay, someone had the viral infection. Let's see if we can properly boost the part of the immune system dealing with the receptor binding domain by using the dimer of the receptor binding domain called Soberana Plus. So it was very interesting because these are data that we published while we are writing the manuscript in collaboration with Ospedale Amedeo di Savoia di Torino. You see, these are people that got the alpha infection, but then they didn't have a lot of antibodies to neutralize it. But when we boost it with the receptor binding domain dimer, you really see an upregulation of the neutralization titers. These are neutralization titers done with live viruses assay. But it was extremely exciting for our perspective was that the population that was infected by alpha variant didn't have almost antibodies against beta. These are convalescent people, because beta had a lot of pretty interesting mutation on the receptor binding domain, unfortunately nicely close to the Omicron one. But when we boost these people with the receptor binding domain dimer, so the vaccine called Soberana Plus, this was actually the upregulation of the neutralization titers. So the strategy actually was correct to present to the immune system the receptor binding domain, offer you maybe the opportunity to recognize part of the antigens that maybe the full spike didn't. And these are safe vaccines, and in addition, also the price that we estimate as a symbolic one is $1 per doses. So, but the most interesting part in our rationale was to develop a vaccine not only for the convalescent people, of course, but for the adult, but mostly for the pediatric population. And what I will describe you is what we call Soberana 2. Actually, it's a conjugate vaccine where different copies of the receptor binding domain are covalently conjugated by a malamidotiol chemistry on a carrier called tetanus toxoid, exploring and exploiting the technology that Finland Institute and other companies have used to develop vaccines against Streptococcus pneumoniae, Neisseria meningitis, Haemophilus influenza type B, etc., etc. So we express again the receptor binding domain in a mammalian cells, and we covalently conjugate it on a protein carrier that is the tetanus toxoid. As you can see here, we can control the amount of receptor binding domain that in general is on average around 6 copies of receptor binding domain per protein, tetanus toxoid. And as I mentioned already, with a technology that has been used already since a long time, using a malamidotiol chemistry. So why we do this type of approach? Because it is known from the field that if you want to optimize the immune response against a small protein, or actually against polysaccharides, that is our field of research, if you conjugate the receptor binding domain to the tetanus toxoid, in somehow, I will try to make it easier, in somehow you're gonna help your B cells to talk to your T cells, and this will increase the quality and quantity of the specific IgG against the receptor binding domain. All this cartoon is extremely exciting when we talk about pediatric vaccines, because to trigger an optimal T-cell responses in children, you must conjugate your antigen to a protein carrier like the tetanus toxoid. So we did a phase 3 trial. We did all the trial. This is an adult population. And against symptomatic COVID-19 disease, we had 92% of efficacy versus placebo. These are 2 doses of the conjugate vaccine. But because in Havana, the beta variant was replaced by the delta variant, so a very bad scenario, epidemiologically and clinically, the vaccination scheme already includes the boost with the receptor binding domain dimer. So as you can see here, these are antibodies against the IgG. These are at time 0. This is after 2 doses, and this is with the boost. And this is in the young population, and this is in the older population. And actually, it's always hard to compare this data around, but it's always elegant to express this by showing a panel of convalescent people that was a panel of Cuban convalescent people that got the virus. So as you can see, starting from 0, we have a pretty interesting and qualitative important immune response. Not only in terms of amount of antibody that we were able to trigger, but also in terms of neutralizing titers. So this is the same panel, and you can see here that the population with the boost, so 2+1, were nicely able to neutralize the virus in vitro with live virus neutralization assay. We recently also start to have data about the duration of these antibodies, although with Delta and Omicron, it's maybe not sure that the amount of antibody do correlate with protection, but of course, having a conjugate vaccines, it also helps in quickly develop these antibodies. So briefly here, you can nicely see here that also in terms of antibodies against the spike, but also in terms of neutralization titers, after after let's say 7 or 8 months, we still have important neutralization ability triggered by these vaccination approaches. But for us, it was extremely important to vaccinate the pediatric population. So Soberana 2 at the moment is the only vaccine in the world authorized below 5 years old and administered to millions of children. In addition, Cuba realized that during the Delta wave, the cases of reinfection of the adult population were mostly driven by the infection in the pediatric population. So we think that vaccinating the pediatric population will decrease the transmission of the virus also in the adult population. And I'm going to show you also some of these data. So nicely here, these are the data that in preprint, now under revision. You do see here that unlikely the mRNA vaccines that they do work better in adults compared to children because of the T cells immunity of children. As you can nicely see here, the children with the full vaccination schemes have higher titers of IgG against the receptor binding domain compared to the adults. Also in terms of neutralization titers. So these are the neutralization titers with live virus neutralization assay in children and here in adults. Of course, I don't have the time to show you. The vaccine is pretty safe. In addition to the children involved in the clinical trial, also the million of children followed by pharmacovigilance services, they show extremely high safety profile. So this is what we think is extremely important. So I didn't compare Cuba and United States for political reasons, although sometimes it's also important to mention that Cuba is a country that is under blockade from United States and not only since already 60 years, but they are very good in developing a public biotech. And these are actually the results. So this was actually the only wave that Cuba had, and that was very dramatic, was the Delta wave in the summer last year. And this is actually comparing USA with Cuba. This is how normalized by population. This is actually how the first and second Omicron wave appeared in Cuba. Because we start vaccinating here at this moment in the middle of the Delta wave. Nowadays, 90% of the Cuban population is vaccinated, 60% of the Cuban population is boosted with 5 different approaches, and only country in the world, 97% of the children population below 2 years old is vaccinated with a conjugate vaccine designed for the pediatric population. And this is actually one of the reasons that maybe will explain how the Omicron wave affected in a gentle way an island like Cuba. I conclude with this approach. There are a lot of inequalities in this world, and this is actually how the vaccination rates is running in low-income countries. This is actually the way high-income countries are overcoming this type of approaches. But it's interesting to show that a poor country, decently poor country like Cuba, that has invested in public biotech since already more than 40 years, proved that you can produce your own vaccine in a public manner. And actually, Cuba at the moment is the country that injected more doses of vaccines for 100 people in all the world. So again, it was an honor for me to be part of this team that I'm co-working with them since 2014, mostly on Streptococcus pneumonia vaccines. And what I try to show you that every time you have to think about a therapeutic agent, that can be a drug, that can be a vaccine, you do have to consider carbohydrates in your analysis. What I showed you here, that when we have decided to select the receptor binding domain as our main antigen for the vaccines, we did know that this receptor binding domain should be expressed in mammalian cells to keep the proper glycosylation for different reasons, but I only mentioned here structural important key reasons. So, well, for the presentation of today, these are all the collaboration, but I have to thank, of course, Fila Institute, especially the director and the directions, especially Professor Vicente Verez Bencomo, a brilliant scientist and a brilliant person that give me the honor years ago to be part of this impressive team. And of course, also all the other collaborators and CNR to hosting me as a permanent researcher since the last 2 years, and also Amsterdam Medical Center, where I'm still affiliated. So thanks a lot for your time. I hope I've used a proper language, and I'm looking forward to having some curiosity, suggestions, etc., etc. Thanks.
Thanks, Fabrizio. Very interesting, very, as you said, far but close at the same time, because everybody touched this quite heavy problem. And so if there are curiosity for Fabrizio immediately now.
Yes, I have a question, Doriano from Trieste.
Please.
I'm very much intrigued about the development of a vaccine focus for children under a certain age. The question is the glycosylation pattern and the composition of the carbohydrate different on age? Let's say adults have a pattern and composition different from children.
So it is a very interesting and fascinating question. So technically, statistically talking, there are not a lot of differences from a viral infection, so a virus that is using our machinery and is expressing its glycans to the host. So from this type of machinery and also for other type of glycoproteins, there are no quantitative massive differences between adults and children. Of course, for any other proteins, maybe there are very tiny differences between my glycoprotein, Cinzia glycoprotein, and Vincent glycoproteins. Okay, so but this is on an extreme level, maybe very, very tiny ones. So statistically talking, as far as we know, there are no differences in the mammalian glycosylation system between adults and children.
So why then this vaccine works so let's say better in children than in adults?
Yes. So I didn't stress on that. Is mostly on the strategy of the conjugate vaccine. So the T cells of children, they really require the tetanus toxoid or other immunogenic carrier. We know that from the bacterial vaccine. If you have a bacterial like Streptococcus pneumoniae, Neisseria meningitis, and you keep the capsule, and you inject the capsule to Fabrizio, Cinzia, and Vincent, you're going to have a weak response, decent for a travel, but not that much. But if you inject this in children, it's not going to work. It's completely silent to the immune system below, we think below 10 years, below 5 years old. But if you conjugate the polysaccharides on a protein carrier, and this is actually what we did with the receptor binding domain, you optimize the crosstalk between the T cells and the B cells. So this is that big. So the main difference is because T cells in children are different, the strength by which they recognize antigen on the TCR is different. So we did optimize this with the conjugation to tetanus toxoid.
Thank you. Thank you.
Fabrizio, would you do you have a question, an open question concerning either the morphology or the structure, atomic structure, I mean, that you still have in your mind unsolved, by which you would like to end your half an hour?
It's more than 1 year that we've tried something and we didn't succeed on that in find the collaborators. There is a lot on the receptor binding domain, there is a lot on the spike, but there is not a lot on the dimer. So what is not clear to us, we have some estimation. If this is the disulfide bond, this is the receptor binding motif, so the one that's really anchoring to the H2. We are not sure if it's like this, it's like that. We don't know that. It's pretty hard to crystallize glycoproteins. Haha, this is why our field are starting to interact because it's very complex, because of the water, because of the mobility. This is what your colleagues are always discussing with me. So yes, there are structural things in the vaccine formulation of Soberana Plus, the dimer. This is something we really don't know. About the conjugate, I know it's already more complex because it's huge protein with 5 different copies on it. So, but if there is an open question that we do have is how to, let's say, what the dream would be to have a crystal of the receptor binding domain dimer expressed in mammalian cells is an open question. It's an open question. We have tried to do it, but unfortunately, I have to say that unfortunately we have tried some collaborations, but then at the moment the head of the dean or whatever, they hear Cuba, they stop answer to our email. We know that we have used to it. I mean, unfortunately, this is also something that we have to add. So all these colleagues, we did all that without any money from the Gates Foundation. And also pretty hard to collaborate. Of course, we have a network of co-workers. Like as a scientist, they like to work with great scientists like my Cuban colleagues. They're just scientists. And yes, so unfortunately, every time we have tried this, of course, it was USA, but there are active collaboration within Cuba and USA. Eh? But in that case, we were not lucky. So yes, this is an open question.
Okay, thanks. Okay, so now let me share the floor with Vincent Olieric from PSI, and he will open the field to synchrotron radiation, and so add some important information for this community. Thanks.
Thank you. Can you see? Does it work? Yeah, it works, right? Yes.
Yes, fine.
Okay, thank you for the invitation. My name is Vincent Olieric. I'm a beamline scientist at the Swiss Light Source at the Polscha Institute in Switzerland. And I'm going to talk a bit about the activity we have in the group, together with some mention about the COVID-related research that has been done and is currently being done. So the synchrotron is close to Zurich, 40 minutes away from Zurich. This is another view from the Swiss Light Source or from the Polscha Institute. The Polscha Institute is around this Aare river, and you can also see the Swiss free electron laser here that has been built. See this old picture still in construction, but since then the free electron laser is in operation. So I will talk to you about the activities of the MX group, some COVID highlights. I choose to present some relevant hardware development because I felt that the audience of this cafe would be more chemical crystallographers. So I will go through some of hardware development we've done along this line. I'm going to talk about some changes that is happening in our field, recent changes, and quickly present some new opportunities for the future, and especially in view of the upgrade of the Swiss light source, which is called SLS2, which will happen sometimes, which should start end of next year, so 2023. Okay, so this is our group. We are a bit more than 20 people, split it in 4 different categories. One is more like based on applications. One is more dedicated to instrumentation. Another part is data group is more software involved. And the last group is involved into the sample preparation. We are in charge of 3 beamlines. PX1 has been designed has been the first one in operation since 2001. It's a non-dilator beamline, very high flux. So all beamlines are tunable. The PX1 beamline, we can change we can make it a microfocus station. I will maybe skip on this part for now. All beamlines are equipped with a sample changer, so we do a lot of automation that I'm going to describe you. PX2 is an industry beamline. This is a copy of PX1 for industry. This is in operation since 2004. And PX3 is the latest one in operation since 2007. So we have, as I said, academic users, industry users, and we are also in charge of a third beamline, which is on the FEL side, so the free electron laser side. And maybe I'm not going to talk too much about this for now. Just to say that the PX3 beamlines is the beamline that I've been in charge for the last 15 years. And we just started last month. Was our very last measurement on this beamline. So this is a picture of the group that is trying to fit inside the mini hutch. So it's a very tiny hutch, but we all managed to fit. So the beamline has started being decommissioned. So here you see the removal of the front end component inside the ring, and the hutch being dismantled. The reason is that we are starting an upgrade for that will last about a year to be back. So right before the upgrade of the ring itself. I may explain this in a couple of slides. So historically, the beamlines at SLS have been deeply involved into a lot of hardware development. This has started 20 years ago with the first with a microfocus station. The Porsche Institute is very famous in hardware detector development with a Pilatus detector, which has been installed at PX1 beamline in 2007. This has a huge impact in the field because of the throughput that the detector brought. So no more shutter to collect the data. The detector is capable of doing shutter-free, shutterless operation. So we went from like 20 minutes data collection to maybe 2 minutes. So it had a huge impact, especially on our industry users. So this kept on going with an Eiger detector and nowadays a Jungfrau detector which has been actually designed initially for the Swiss PEAR operation. So microspectroscopy development, some hardware development towards multi-axis goniometers that I will show you. Some more exotic type of experiment with crystal being levitated or time resolved, and more recently rastering, or like here you see a picture of TEL. The TEL is our sample changer, which allow now to do full remote operation and even unattended operations. So a lot more experiments are being done these days, fully unattended. That means the user sends samples and we measure them. We measure for them. This is collected automatically. So this last development here was very crucial to still maintain operation during the COVID pandemic. So 2 years ago, so March 2020, this is a picture of our hatches, the 3 beamlines. They became all empty, where we used to have users coming. So here is just a little graph of the remote operation at 2 of the beamlines, PX1 and PX3. So the remote operation was ramping up. We were starting to be, I think we were ready at the right time, because when the COVID hit us, then we were ready to operate 100% remote, and there was no way we could operate differently. So this automation, this sample changer, was crucial to operate. So our users typically prepare crystals in this type of litho loops, or sometimes nylon loops, that stick on at the end of a pin like this. These pins are stored into a puck that is maintained under liquid nitrogen temperature. This is the enclosure for the puck. Then several pucks can be mounted into a canister, and the canister, this is 7 pucks. This will go into a DWAR. Here you see 4 different DWARs. That's what the user sent to the synchrotron in a frozen state. So then we mount them with our sample changer. So this is a view of the PX1 station with a sample changer unloading and loading a next sample. So this operation is less than 20 seconds, and so users with this can go at a throughput of maybe up to 24 samples per hour. That's how fast we collect. At least the fastest users that are coming from industry. This is another view. If you were to mount your sample from at ambient temperature, room temperature, so we have a little chamber from where the samples can be picked up. So the sample is mounted on a goniometer here. The crystal would be sticking here on the pointer at the end of this pin. The X-ray are coming from the right side. Here is a beam stop to collect the direct beam, and here we collect our diffraction pattern on a detector, like here on the Pilatus at PX3. So during COVID, we went to 100% remote operation, and we also opened a special call for COVID research that I'm going to show you now. Just a few highlights. One was very much along the line with Fabrizio, what Fabrizio presented, like targeting the spike. So these are nanobodies that are attached to the RBD actually. So the RBD can interact with 2 different kind of antibodies. They are produced from llamas, and so this is a way to stop the interaction with the host. So this act, if we see at the infection cycle, this is acting at the very first step, which is attachment of the virus against with the receptor ACE2. So again, if you can kill this interaction, you have a very strong antiviral drug. Another Another highlight was more like down at the once a virus is incorporated into the cell during the translation of lots of what we call non-structural proteins. So one of them, I just want to mention it because again, it was like the beginning of the pandemic. Everything was locked down. We opened this call. So on March 29, this group from Frankfurt submitted a proposal. There was a shutdown that was cancelled to actually accommodate this beam time. And a month after the submission of the proposal, the paper was under review and got published a bit later into Nature. So that was actually a very, very, very quick actions that were taken here from this group. So since then, many, I mean, pretty much all the genome has been from the SARS-CoV has been resolved or has at least a model. So this is then some. Let me see. Yeah. So some of these targets have been studied by a method called fragment-based drug design, which is a way to identify lead compounds on a target. So basically, what is done is that by crystallography, we screen a library of compound, a library of ligands that we soak on our crystal. So this can be as big as like 1,000 ligands. So we harvest them all, we collect them all automatically, and automatically process, and with a goal of identifying hits that may be then used to, I mean, in a way to bridge and to make a more compound that is with more affinity. So usually you screen against a library of, let's say, 1,000 fragments. Maybe if you see, you're happy if you see 10 of these ligands bound to a structure, with a hope to end up with a drug. So this is some of these experiments that are currently ongoing at our crystallization facility. And now I want to switch to talk to you about some relevant development that we've done at our beamlines, especially some that are more relevant for chemical crystallography. Among those are the detectors. So I already talked to you about that PSI was deeply involved into developing detectors. Most of them are now commercialized by a very successful company called Dectris. So this started with a Pilatus in 2007. Then now the Eiger generation, and currently we are testing Jungfrau, which is capable of like very, very high frame rate, which enable like very, very fast data collection. And so one key thing for chemical crystallography is a detector distance, being able to come very, very close. So if the sample would be here, in some instance, we can come very, very close here with a detector, or even have a tilt that would allow the very high angle, catching the very high angle reflections. Some incremental, yeah. Is there a problem?
Listen to you, huh? No problem.
Okay, good. So the Prigo goniometer is a multi-axis goniometer that is a way to reorient the crystal in the beam and to put some reflections that would not be into diffraction condition into diffraction condition. This has been now commercialized by a company called Smargon, and I just want to show you how automatic multi-orientation data collection would look like. Almost, I mean, that nowadays, this is how fast we can collect data pretty much. So that would be a rotation of 360 degrees at a speed of 180 degrees per second with reorientation and another measurement being done. So basically, so this was also key in some experiment we've been doing in the past with regard to experimental phasing. Another piece of instrumentation that I want to present here is something that has been developed in Japan. It's a deep UV laser, so that crystal can be shaped to any shape, any kind of shape. So they have like dedicated instrument that you can see here. And basically what happens is that you can get rid of like extra part of the crystal and like make your crystal with a spherical shape, like to control the absorption better. So that is especially important when you do a low energy experiment. So with about chemical crystallography, we've been, so users do use it routinely, especially industry. So they benefit from tunability, beamline is tunable, high flux, a multi-axis goniometer, as I said. Sometimes the microbeam, the large detector, is also quite important. And we can change the sample with a robot nowadays, even at room temperature. And we have had some highlights. So a group from Innsbruck identifying new minerals. So they came and identify about 15 new minerals at our beamlines. And I want to mention the collaboration that I'm having with Anna, Molitani, and Cynthia on the perovskite material for solar cell application. So last year was a very productive year. So all of this development that I just presented, so the goniometer, detector, and laser shaping technology has been very, I mean, we've developed this for the purpose of doing experimental phasing with a method called SAD or MAD. So that means de novo structure determination. But this is this has been like this is very much changing. The field, our field, is very much changing. So I want to just spend like a couple of slides with what is happening in our field now. You may have heard of this the breakthrough of the year was last year. So artificial intelligence used to solve the folding problem. So it's called Google DeepMind AlphaFold. So AlphaFold program can now predict with experimental accuracy most of the structures. So by just giving the amino acid sequence. So for proteins, you can just put your amino acid sequence. So I'm going to skip this. You put your amino acid sequence into this program, either directly on the web with AlphaFold Collab, or by downloading the source code of the source code, or directly searching into the EMBL database, and you may end up already with a very good prediction of your protein. So this is just one example which we solved happily with by experimental phasing, where at the time when we had no homologs. So last summer, AlphaFold came into play, put the sequence into the program, and it ended up like fitting extremely well. So this is just one example out of many, but this is really to say that the molecular placement now, this way of solving structure with the AlphaFold prediction, is clearly replacing experimental phasing for proteins. So in the case of Fabrizio, maybe I'm wondering, there is another version of AlphaFold called AlphaFold Multimer, where there will be a chance to actually predict the how this dimer could assemble. So maybe something to do to try. Another revolution, another changes are happening in our field is the advent of the cryo-electron microscopy technology, and how nowadays it can really access resolution on very big complexes, and so the number of model is growing exponentially. This is to compare with the X-ray models. That is a bit. I mean, it's not going anymore exponentially. That's very clear. But cryo-EM is very developing. I just went through. I just want to show you one example that I saw this week that was really stunning to me, like this cilia. So I've been involved a bit into studies on microtubules, but this like really this is what you can do nowadays on very big complexes. I'm not going to spend too much time now. So experimental structural determination in combination with AlphaFold is now can be done easily more easier with MX and EM. So AlphaFold has solved the phase problem. So what is left for MX? And that's going to be the last part of my presentation. Just quickly show you some of what can still be done by crystallography, and with some opportunities for the SLS2 upgrade. So it's still possible to visualize some of the heavier atoms, like heavier atoms, like for DNA. In the case of DNA, like you could spot your phosphate backbone in the case of DNA here. It could still be good to combine with some spectroscopy tools to sometimes correct the misassignment of some ions, like in the case of that structure, Cas9, where manganese atoms were wrongly modeled when it should be potassium. Identifying potassium channels is another example of visualizing hydrophobic tunnel in the case of these studies with xenon. So there is still some experiment that can be done with to use anomalous signal. Otherwise, what is currently being like done is like some different crystal delivery with jets, with sample directly in mesh or directly in the crystallization container in situ, and this has led to some new new thing we can do at the beamline, like rastering very quickly meshes that contains crystal to identify where they are located. So this would be because of the very fast detectors that we have, we can screen very quickly through large sections. So this is this would be a way to identify crystals. It's going to show up like this. Yeah. So sample delivered with jet. This I'm not going to go into too much detail. This is all has been developed with like common development for FEL applications, or even like crystal being in levitation. So this is at room temperature. So this would be a good way to collect and inject, like for instance, like ligands, and to find out how the ligands will interact with the structure. Okay, time resolve experiment can be also temperature resolve. So we've done a study here where by changing the temperature we could see that one of the ligand would really go from one side to the next. So I have a little video here that it plays, and if you focus here, you see that this part disappears and this part will appear toward the end of the data set. So really, the ligands completely flipped and with a ramping temperature. So what is the SLS2 upgrade? The machine upgrade is a new lattice and will lead to a beam that will be much sharper, so lower emittance, so very small beam and more flux, more parallel. So we will gain for all beamlines. And so the operation of the 3 beamlines has been done like this: 2000, 2004, 2007, and we are now currently already upgrading the PX3 beamline prior to the SLS2 upgrade in a way to prepare to spread the work over a longer time, and also to be ready and test new technologies that will be employed at the other beamlines. So this is where we are. We have started the upgrade of PX3. We hope to be back in operation so in about a year, right before the real upgrade of the machine that will last in the entire 2024 and part of 2025. So quite a long shutdown. But hopefully we'll be back with three better beamline. So PX1 to discover new, to apply more serial crystallography approach. PX2 will be a true microfocus for industry, and PX3 will be a more of a, conventional experiment, but capable of microfocusing and like high throughput experiment. So that's what I want to present you for this afternoon. Honestly, thank you for your attention. So this group, I've been like presenting you the activities of my group, of our group. I want also to thank some, academics, users, and industry partners. And yeah, I hope I haven't been too long, and I'm happy to take questions or to
in time. Yeah? Really half an hour, perfect.
All
okay.
Right.
So very nice and complete trip in your world. So if there are questions for Vincent. Maybe from Fabrizio, some curiosity.
From my side, it's just to, have to really understand if this is possible for the dimer of the receptor binding domain, but I will write you. Let me first understand what we can do, what is possible, because we have the material, so let's see. At least a try. It's gonna be interesting. The community will need a boost for a lot of time. So also understanding structurally what we have there and why maybe it's more interesting than other approaches is interesting.
I had a question for you. The model that you showed with all the carbohydrates, how is it generated?
This has been done by other colleagues in US. These are publications with a mix between dynamic studies.
That's all. So that's all. Yeah, it's a pure model based on molecular simulation, or
yes, molecular simulation and also some cryo-M at some point. But now I don't remember which one, what one. But it's pretty hard to have. So about the crystal, it's difficult. And yes, there is also something with the monoclonal, with the receptor binding domain, with the proper glycosylation, and some monoclonal, but I don't remember what has been done in terms of crystallography. I will check because I forgot. I mean, I'm stopped to follow this part of the literature like last year, but for sure about the dimer, we are the only one that we have it. So also in terms of a product, so it's quite unique. But I will send you an email with the 3 key publications that we use in the field, and I will give you the characteristic of the dimer, and let's see.
I had a question about this. So did I get it right that the RBD is less covered by the carbohydrate? It's like more exposed?
The RBD is less covered, but carbohydrates are there. The point is that forgot about the carbohydrates. In any case, this is the receptor binding domain. Imagine that there is a third one, but they only have 2 hands. When H2 is coming, this is going up. Okay, so we do assume that in this part you have antigen that maybe can be interesting. In addition, there's a glycan here that is pushing this one up. So this part is pretty interesting also because polyclonal antibodies, monoclonal antibodies that are also able to recognize alpha, beta, but also omicron simultaneously, they do recognize these, let's say, glycopeptides, let's say, this part. So the RBD is the most exposed part of the spike, yes. The RBD, it is glycosylated, but not, let's say, completely masked like HAV-GP120. So yes, it looks like a fully ball of sugar, nothing to compare like GP120 of HIV. In that case, it's almost impossible to reach the protein site and the antigen. So likely this virus is not playing that well. Maybe glycans are playing other roles, other entry route, or also keeping the immune system a little bit down to let's say, to say it easy. But yes, this is what we know.
And in terms of structure, you said you would like to know how if it interacts like this or like this, right?
Yeah, because it's a disulfide. So here is disulfide bond. So in any immunological interaction, what we call is not a multivalent system, but is a dimeric system. So two is already better than one, but also because let's see, the immune system is looking at these two simultaneously. So, but we don't know energetically what is most favorable, and because this can explain why this is also pretty interesting. Because we speculate that it's something I don't know in terms of repulsion what is going to happen and why, but this is a question. So what is presented to the immune system? Is something like this? Is something like that? Because this can be also interesting to understand how other vaccines can work when you have a small protein. So now we know you have a small protein. Likely we could have the dimer because it's a piece of serendipity. The same dimer, you can reduce it and conjugate it on tetanus toxoid. So imagine for production is one pipeline. From one fermentator, you have the pipeline to two different products. That is also pretty convenient. So it's really, from a pharmaceutical perspective and an immunological perspective, it's pretty interesting to understand structurally what we have in our hands. And it's not the limit. It's not the full spike. It's just the dimer of the receptor binding domain. From your perspective, it's extremely complex, but it can be a starting point. And material is, of course, you don't have micrograms. If you need material to try any condition, or this is a product of a pharma company, so we have amount.
May I have a suggestion? I mean, it reminds me a similar problem with a full-length immunoglobulin is how the heavy chain is oriented comparing to the light chain, the FC, and so that has been addressed in several times, also for for checking the homogeneity for pharmaceutical application by small angle X-ray scattering. So I think to have a rough idea of if it's an homogeneous solution, because you might have an average at the end. You don't need, not necessarily you might have a trans or cis orientation. Let's make it simple. And but definitely probably a Sachs measurement will tell you on the envelope. Because if this fully cis or fully trans, they are quite different. They should be able to be distinguished by small angle X-ray scattering. Without going at the first place to any crystallization on anything, just to have a rough idea of the envelope of the dimer. The problem probably is that the disulfide being exposed can be easily oxidized. And therefore you end up with the monomers. On the other hand, you might use some mercato heptanol, but that is not good for Sachs measurements. But anyway, I mean, there are experts. So.
Just to conclude, just a very quick question to Vincent for this upgrade. So, do you promise to study 1 micron small crystals, right? I mean, the minimum size is that, right?
This will be, I mean, definitely should be able to do that. Yeah.
And combined with levitation?
So that would be a slurry of crystals. I guess, I mean, combined with levitation, the thing levitation is still rather, it's quite a big drop. The drop is still not, the crystal will not be, okay, the crystals still have to be within a drop, and the drop will be definitely bigger than 1 micron. So maybe this will not be the right media or the right way of collecting with such a small crystals. This would be more, if you have 1 micron crystal, it would be more like the fixed target where you raster scan, or something with a slurry of crystal in so you shoot through a jet, whether it's like something viscous or like a liquid jet. But probably not with acoustic levitation.
Sorry, Cinzia, can I?
Yeah.
Sorry, Cinzia Drayton. Yeah. Can I say just a few comments?
Yeah, sure.
Yeah. Okay. No, was just about what Fabrizio said before about using dynamic simulation and cryo-EM. I guess that probably you use first cryo-EM, and after to fit better the model, probably you use dynamic simulation, so to improve your model, I guess, first of all. And the second, it's that I agree with Doriano to using small angle scattering, and probably even in that case, you can use both combination of Sachs with cryo-EM as well. So if it's possible for you to do cryo-EM, you can do first Sachs, having a rough idea of the envelope, and after looking inside a cryo-EM map. Okay. Anyway, thank you very much, Bob, for your presentation. Very nice.
Thanks a lot. Thanks a lot for the suggestion. I took some notes.
Okay, so thanks, and I think the time is over, and let me clap my hands for this very nice cafe, strong cafe. Okay, so I hope we will continue afterwards as well. So
thanks a lot.
Enjoy. Grazie a tutti.
Thank you.
Bye. Thank you. Bye. Ciao.
Ciao.